Okay, so this is essentially a walkthrough of what we are going to do in the Bacterial Transformation Lab. So we're going to start with some E. coli cells, and essentially we're using E. coli because it grows really quickly. Sorry, let me lock the screen there. It grows very quickly, and it's just easy to use, easy to grow. So inside these E. coli cells, they have their DNA, their chromosome. So they have a single chromosome. They also have what we call plasmids, which we've discussed plasmids in class. These are just those circular DNA pieces. They usually only code for 2 or 3 genes. But they're just part of the bacterial genetic information. So we are going to take essentially our own plasmid. Okay, so we'll call this our plasmid. And this is what we're going to actually transform and put into our E. coli cells. So first, on our plasmid, we want to talk about a few different genes that are on the plasmid that we're going to transform. So the first one, I'll just kind of label this little section here. This is an ARA gene. It's essentially an arabinose gene. What it does is it allows the bacteria to break down arabinose. Okay, and arabinose is just a sugar, right? It's just a type of sugar that the bacteria is going to break down. We also have a second gene on here. We call this one GFP, and it essentially just codes for a green fluorescent protein. So this is going to essentially glow under UV light. Okay, so this is the plasmid is going to give the bacteria the ability, this gene here, to glow under UV light. This gene, okay, it's a special, kind of has a special function. We call this an inducible gene, right? And what we mean by inducible is that something has to induce it. Something has to start it. So if you think about, if you've probably heard this term before, like inducing labor, that means like getting it started. So something has to actually turn on this gene, and what's going to turn it on is arabinose. So the whole function of this arabinose gene is to turn on GFP, right? So what that means is when arabinose is present, this gene is going to break it down, and that's going to turn on GFP. Okay, so maybe I should say when arabinose is present. Okay, if arabinose isn't present, then there's nothing to break down, and it won't turn on this gene. Okay, the last part of our plasmid, we actually have this other section here. Carbon, I might be spelling this wrong. I think it's actually the other way, but carbonicillin. This is a carbonicillin resistance gene. Okay, and when you say carbonicillin, that kind of sounds like penicillin, and it is. It's a penicillin-like antibiotic. And if we know what antibiotics do, we know that they can kill bacteria. So this gene is going to essentially mean that if it's exposed to carbonicillin, it will not affect our bacteria at all. Okay, so there's these 3 genes. This gene is always present. this gene does not have to be induced. This is called constitutive. Essentially, it just means it's always on. Okay, so we'll say always on. Right, so we have 3 genes. One, green fluorescent protein allows the bacteria to grow, to glow, as long as it has the arabinose present, because that has to turn on that gene. And then we also have the carbonicillin resistant gene. Okay, so essentially in the procedure, okay, we're going to set up 8 different plates. All of these plates have something that we call LB. This is just media, okay, nutrients that E. coli really, really like. So essentially, if LB is present, our bacteria will want to grow. E. coli will want to grow because it's just nutrient-rich media, essentially, like a growth media. There's also a plate that has LB and arabinose. Okay, so I've added that extra sugar in there. We also have a plate that has the LB, right, which is just the nutrients, with the carbonicillin antibiotic. And then we have this last one is this combo. So we have arabinose and the carbonicillin on this plate, all right? So this LB plate, these are essentially like our control, right? We would expect our bacteria to grow no matter what, because it's just pure nutrient broth. So it's just a really great environment for bacteria to grow. I'm not changing anything about it. I'm not adding anything special. So our bacteria should grow like crazy on these control plates, okay? And then if you noticed over here, right, we have like these plates will all be incubated and inoculated with bacteria that we gave the plasmid. These plates have no plasmid bacteria. So these are just regular old E. coli cells. Nothing has our plasmid. So it will have none of this. None of these genes will be present on these plates down here with these bacteria. Okay, so this is just plain E. coli. So what I want to do is kind of be thinking about, what do I expect to see on these plates? So like we said, these LB plates, these are our controls. So I would expect to see lots of growth, right? Positive growth. Because these are just regular nutrient-rich plates. My next plate, right, I have just my LB media, so just nutrients, and I have arabinose. So do I expect it to grow here, right? I mean, I think so. We're not doing anything that would keep bacteria from growing, right? It doesn't have any type of antibiotic, right, that's going to stop bacteria from growing. So I would say, yes, this should have some growth in it, right? Now we're over here with our carbonicillin. So remember, carbonicillin is an antibiotic. So would I expect my E. coli to grow in the presence of antibiotic? So I have to start thinking now, well, if I gave it the plasmid, right, that means I gave it carbonicillin resistance. So yes, I would expect to see growth here, because this E. coli with the plasmid is resistant to that antibiotic. So I should see it grow even though there is an antibiotic down here. However, with my bacteria that doesn't have the plasmid, it doesn't have any resistance to that antibiotic. So I wouldn't expect it to grow, okay? Anytime it has that carbonicillin. So just like down here. Okay, it has that carbonicillin. Doesn't matter that arabinose is there. I have an antibiotic. I don't expect it to grow. Up here, this plasmid, right? Yes, I should have growth, because I have that resistance to my antibiotic. The second part of what we want to look for is, will it glow? So I'm looking, are we going to see this fluorescent protein causing it to glow, right? So we have to think, what is going to allow that to glow? Remember, I said I have to have arabinose to turn on that gene. So when I look down here, right, I know that my negative plasmid, my bacteria with no plasmid, all of this should have no glow because it doesn't even have the gene. However, with my bacteria with the plasmid, right, I would expect some of that to glow because it has the gene. So I just have to think, okay, well, what is going to trigger it to glow? Arabinose. So any plate that has arabinose, right, I should see some glow. All right, so when we come in the next day to look at our plates, this is what we should expect to see. If we don't see this, we want to kind of start thinking, what could have gone wrong in my procedure? Because remember, there were lots of steps. It was very meticulous. You had to add just the right amount. You had to make sure it was mixed well. One of the steps was heat shocking it to cause that plasmid to actually be absorbed into the membrane. When we heat shock it, it actually causes E. coli to open up its kind of cell membrane transports. And so this plasmid, all it has to do is just sneak in there while those are open. And then once we put it right back on ice, it causes all of these to just close right back up, okay? Which means that it's now trapped our plasmid inside with the other original plasmids. Okay, so our goal is to have as many cells as possible to absorb that plasmid. So the more cells that absorbed that plasmid in our heat shock, then the more growth and glow we should see down here in our results, right? If we're not seeing a lot of glowing, right, we may have growth on some of these, right, that don't have any kind of antibiotic in it. We may see a lot of growth, but if we're not seeing a lot of glow, that means that we didn't get a good transformation because we didn't get a lot of cells to actually absorb that or take in that plasmid. Okay, so we want to kind of be thinking, well, what could have gone wrong? What could I have messed up? So when we come back together, we will look at our results and kind of see how